Right.
Hello. So in case you are trying to use the virtual microscope activity and it doesn't seem to be working on your computer, at least you can view this video and follow along. Maybe pause and replay some parts if there's something you need to see again. I'm just going to follow through the instructions that were posted on D2O and let you see what's going on the screen. The first step says click on the Explore link at the bottom of the page. Then we want to click on the question mark on the slide box. So this is the slide box. Click on the letter E slide. Okay, that's in sample slides. And then letter E. When the microscope view opens, make sure that the 4x circle is highlighted in blue, which it is. So this represents using the 4x objective lens, which is the shortest one or the one with the longest working distance. It has a magnification power of 4x, plus we've got a 10x objective lens. So everything we're seeing is a total of 40 times larger than it would really be. So then we want to use the slider to coarse focus to find the letter E on here. So I'm using this slider here, and I want to do this until it looks sharp. I think that's overshot it. That looks pretty good to me. Then we want to use the fine focus to make it crisp and clear if there's any way we can improve it. On an actual microscope, you would be looking at the knobs that are here. The big one on the outside is the coarse focus. The one that's the smaller one on the inside is the fine focus, and they're geared so that they turn together. And the gear for the coarse focus just has fewer teeth than the fine focus one. So when you turn it, it moves more per turn. You don't need to know that for this class, but if you're ever using a microscope, that's useful information. All right, that looks about as focused as I think we're going to get it. You can click on the E in the viewing window to move the image and visualize different parts. It says to sketch your view of the letter E in the 4x area, or at 4x in the results area. So you're not turning anything in, but I do want you to make note. Is this what a letter E looks like when you put it down on a piece of paper? No, it's different. Pay attention to what way it's different. Is it right side up, upside down? Is it backwards? Is it forwards? Is it some combination of those things? That's useful information as well. And yeah, this represents moving the stage around when I click it and drag it. On the stage, we have this slide mover thing. And then when the slide mover moves around, it moves this black part on the screen, as well as this silver part that represents part of the microscope that moves slide around relative to the objective lens, so that you can look at different parts of the slide. Say you had something way over here on the edge of the slide, you could just move it. Now, those are usually on a microscope handled using these little knobs down here. Then the next thing we're supposed to do is click on the 10x circle. Notice that the nose piece on the microscope is going to rotate automatically. So watch, I click here on this 10x and watch the microscope. So that little animation it just did there where it kind of disappeared and then all of a sudden it was zoomed in more. That is sort of how it looks when you look through a microscope, right? As you move the lens away from being in use, it sort of clicks into place a little bit. And when it clicks away out of place, it's like being at the eye doctor when they say one or two, and you know how there's like this period of moment where there's you can't see anything. It's because that you're not actually looking through a second lens, so the light goes in through the eyepiece and kind of stops. Okay, so now we're looking at it at a 10x objective and a 10x ocular. So now this is 100 times bigger than that little letter E. So notice how much of the letter E can you see at one time, right? Before at the 4x, we could see the whole thing, but now at the 10x, we can't see the whole thing, and we can move around a little to see the whole thing eventually, but it doesn't all fit in one screen, right? So no, that means that your field of view has gotten smaller as you've zoomed in. So it says to sketch it, but you don't have to. Just make note of how much less of it you can see. Also, did you notice that when we switched back and forth from the 4x to the 10x, that we didn't really need to adjust any of the focusing? That's because these lenses are designed so that they all focus together. So when you have it adjusted for really good clarity on one, you shouldn't really have to do a whole lot of adjusting in between each of the objectives. Now, in practice, it's not quite this perfect, but it's really darn close. Okay, so now we want to go to the 40x. So now with the 40x objective and the 10x ocular lens, we're now at 400 times total magnification. So what we're seeing in this image here is 400 times bigger than that little itty bitty magazine cutout of a letter E or a newspaper cutout of a letter E. So notice now that it's really kind of, the white area is very dark. So as you go up in microscope objective lens power, you end up needing to adjust the light to let more light in. And that's because every time you're zooming in on a smaller and smaller part of the slide to make it look bigger, you're going to end up letting less and less light in because it's a smaller opening. So the diaphragm down here, you can't see it because it's under this stage here. This is the light here and it shines. And then there's an opening down here that looks like, if you've ever seen an old-timey camera shutter, or if you know anything about how the human eye works, it would be like the iris of your eye. It can open and close to allow more and less light through. So when we slide this light adjuster, you can see that there is some rotation happening below the lens or below the stage of the microscope. So I'm going to adjust it so that the white looks white and the black looks black. But now look, we've got so much zoom in that I don't think there's any way you would know this was the letter E, right? That you can just see just a small part of it. So now if we wanted to go up to the 100x objective, which would be 1,000 times bigger, we can. But this is the oil immersion lens. So see, because it's the oil immersion lens, it's going to prompt us to add immersion oil to our microscope slide. The reason that we need to do that is that the objective lens that is a 100x, it has such a small opening that it can't let enough light in. So we need to bridge that gap with the oil between the slide and the objective lens. The other thing is it's got a real short working distance, which means it basically needs to be touching the thing it's looking at. So this oil is also going to help bridge that gap there too. You put the droplets of oil on the slide, you rotate the objective lens in place, and what happens is the oil, it's kind of viscous and it just kind of like sucks up onto the objective lens. Like if you've ever tried to pick up a droplet of water with your finger, the oil is gonna do that. And the reason we use specifically immersion oil is it's got a refractive index that's the same as glass. Now, what that means is that when light goes through it, and you don't have to understand what this means, but I want to explain it. The refractive index is how much it bends the light when the light beam goes through it. So if you've ever looked at a spoon in a glass of water, sometimes it looks like the spoon is broken or bent because the light is bending differently when it goes through air versus when it goes in water. Well, we don't want that effect to happen in a microscope. So we use this immersion oil that bends the light in exactly the same angle as the microscope slide and the lenses that are both made of glass. And that will avoid us having that bent spoon sort of illusion going on. So I clicked on the immersion oil, and what it did, you see that now there's like this little blue droplet. Immersion oil is really not blue. They just made it look like that so you could tell something happened. And now we're looking at it. And if you see here, this is kind of gray instead of white. So I'm going to adjust the light up some more. Typically, you want to adjust the light up a little more each time you go up in power. Sometimes you don't have to adjust it between the 4 and the 10x very much at all. It just kind of depends on the transparency of the thing you're looking at on the slide. But now, you see, like, we can't hardly see anything of the letter E. As we pan around, it's just black and white because we're looking at such a tiny area at once. All right, so now that we've looked at it under all of these, we want to, when you visualize the letter E slide, click on Remove Slide. So I'm going to click this Remove Slide button here. All right, now it says, because you finished using the immersion oil slide, or the oil immersion slide, I'm sorry, I can't talk today. Now that you're done using the oil immersion lens, you need to wipe that oil off of the lens and clean it because it'll gunk it up and like get nasty. So we're going to click on this lens paper here. And those are called chem wipes. So they're calling them chem wipes to avoid using a brand name. A lot of times people call them Kim wipes, like as in Kimberly Clark brand. But it's kind of like how facial tissues get called Kleenex, even if they're not Kleenex brand. So it's a special paper that's meant not to scratch the lenses, but it will absorb all of that oil off. So we're going to click Done. Oh, they want you to use this lens paper here. We use the Kim Wipes brand ones in our lab, but they wanted you to use the lens paper instead. All right, so now that you've wiped off the microscope, now you want to open the slide box and you can choose any other slide you wanted to look at. So just for the fun of it, let's have a look at some plant cells. Let's do plant cells. All right, we're at 4x objective and we're going to focus it. Hey, look at that. Looks like little rectangles. We're going to find focus it. And let's go up to the 10x. So now here we're at the 10x and you can see these are the cell walls, these straight lines. These are the outsides of individual cells. So like this would be a cell, this would be a cell, this would be a cell, and the dark areas in between are the areas in between the cells. We go up one more, adjust our light up a little bit. So now what we're looking at in these cells is you see this thing here in the middle. This is the cell wall here. This thing here in the middle, that's the nucleus of the cell. That's like, you remember, if you ever took a class where you talked about the parts of a cell, that's where the DNA is stored. And then like all these little green polka dotty looking things, those are the chloroplasts in the cell. So that's kind of neat. That's how the cell makes its sugar from sunlight. Let's remove the slide. Let's have a look maybe at, let's see. Let's look at some fish cells. So these are cells from a fish embryo. And it says white fish, so I'm thinking that that's like cod or haddock, perhaps. The 10x. Turn the light up. And now, because I'm on the 10x, you notice I'm not going to touch that coarse focus again. I'm just going to do the fine until it looks real sharp. Honestly, it was fine before, but. So now that you notice this one, the cell membrane of these animal cells, it's not so square and boxy. It's a lot closer to round and circular. And we don't see any of those little green polka dots because, I mean, fish don't produce their own food from sunlight. I mean, I think you probably knew that. But we do still see a big nucleus in the middle where the DNA would be stored. And then let's try some bacteria slides because, you know, bacteria is going to make you need to use the 100x objective, which you wouldn't see otherwise. So let's adjust our coarse focus. Now we just see little speckles here, right? That's because the bacteria are just so much smaller than any of the other things we've been looking at. This is probably all the better you're going to be able to focus something if you're looking at bacteria. Now we go up to the 10x. All right, now maybe we can focus this a little better. Those speckles are slightly larger. All right, but you notice you can't really see any detail here. Let's go to 40x. All right, starting to get somewhere here. Let's adjust the light. And let's focus in a little more. So now you're starting to see, okay, here's some of these little pink things, and here's some like bluish purple things, but it's kind of still hard to see what's going on here. All right, so now we've got our oil immersion lens, so we need our oil. All right, now we can open the condenser and get way more light here. And we can adjust the fine focus until this little dot here looks nice and crisp. Hey, look, now those things that look like just tiny little blobs and you couldn't really see their shape, now you can see that like those purple things, they've got sort of a stick shape to them. And these pink ones, they're like little clusters of circles. It's like 1, 2, 3, 4 of them stuck together there. There's 2, there's 3. As we look around the slide, it doesn't want to show us the entire slide, but that's okay. It's enough to give you an idea that like how much smaller these things are than plant and animal cells. All right, so we're going to remove our slide. We're going to clean with the lens paper. Even though in our lab we do use the chem wipes, or we call them Kim wipes because they're Kimberly-Clark brand. That's it for the microscope simulation. Hope this helps you. You should have everything you need to take the quiz now.